I want to start off by just giving people a sense for what the tree of life actually looks like. So, can you tell us a little bit about what the major branches of the tree of life are and sort of how big and how diverse each one is? Like how diverse are bacteria compared to animals, multicellular organisms, and so on and so forth?
Well, the Tree of Life has undergone quite a transition since I started at the University of Illinois. I was there at an optimal time, actually. One of my colleagues was a guy named Carl Woese, and Carl figured out how to sequence RNAs, and in particular ribosomal RNAs, and he used that to attempt to develop the first deep branching form of a tree of life, and of course discovered this big split between various prokaryotic lineages. In particular, one group that became known as the archaebacteria, and another at the time was known as the eubacteria. And so suddenly there is this two domain of life tree. Before, all prokaryotes were just lumped into what was thought as one big phylogenetic group. Of course, this I should go back. I shouldn't call it the two domain of life. There were two domains of prokaryotes. The third domain were us eukaryotes. There's a big question of where the eukaryotic lineage fell in the tree. Was it outside of all prokaryotes? I think that was the natural inclination initially, or was it derived from inside the bacteria or the archaea? Was it a mosaic and so on? A lot's changed since that. This was a revolutionary finding, the archaebacteria, of course, but now that we're sequencing whole genomes and get a much broader assessment than just the ribosomal RNAs, it's pretty clear that we eukaryotes are derived archaea.
Hmm.
So now we've gone back to a two domain of life. The two domains are no longer, though, prokaryotes versus eukaryotes. It's the bacteria on one side of what we think is the root of the tree, and on the other side are the archaea. And essentially, we eukaryotes are derived archaea. We're one branch within the
Hmm.
archaeal lineage. So
Okay,
that's
so
the big picture. Yeah.
so at one point we kind of thought there's simple organisms like bacteria, and then there's complicated organisms that include us and some other things. And then eventually we figured out there's actually two sort of completely different types of simple single-celled organisms. And then there's the complex organisms, and we'll get into that a little bit in more detail later. But what we figured out, it sounds like, is that we didn't sort of evolve independently of those two. We sort of emerged, eukaryotes, complex organisms, emerged from one of those types of simple bacteria.
Yeah, that's the prevailing idea now, and it looks pretty solid. Um, it's fair to say we're a mosaic, though, in the sense that somewhere below the root of of eukaryotes, the archaeal lineage that led to us picked up endosymbiont, now
Mm,
called the mitochondrion. And that endosymbiont was pretty clearly derived from bacteria. We've known that for quite a long time.
I see. So one type of single-celled organism sort of absorbed another completely different type.
That's right. And many of the genes inside the mitochondria were transferred to what is now the nuclear genome of eukaryotes. So we're really a mosaic of the two major uh, prokaryotic domains of life in some sense. There's still some debate about, the timing of events here and so on, but um, this seems to be the really broad picture at this point in time.
Mm-hmm. And
No pun
so,
intended.
so so you've got these sort of three big branches of life, two types of prokaryotes, which I'll just call simple organisms, and then you've got the branch of complex organisms called eukaryotes. How dense are each of these branches? Are there way more bacteria and simple organisms than there are eukaryotes and complex organisms like us, or is it comparable, or how do we start to assess that?
Well, that's a good question, and it's partly unresolved. How dense? That sort of is asking how many species there are and so on. Um, and we're getting more of a glimpse of that because we can do environmental sequencing of DNA where we don't even really look at the organism. Many prokaryotes are uncultivatable. I guess the bigger question is how deep are the branches
Mm.
in the different domains? And it's pretty clear now that if you just think about the depth of all the branches and diversity in the entire tree of life, most of it is in fact prokaryotic. Um, Even within, as I said, we're just one branch in the Archaebacteria, which contains many deeply divided lineages. So we're one branch and one side of the root of the tree of life. Most of us eukaryotes are unicellular. And so there's many deep branches of unicellular eukaryotes, as though there was a quick radiation of the eukaryotic lineages. And then a small number of us decided to go down the road of multicellularity. Metazoans or animals, of course, are one of the more prominent ones, and land plants and other fungi to a certain extent. But us animals are a very tiny part of the branch of the entire eukaryotic tree of life, and mammals an even tinier speck on the tree. So most biologists, even evolutionary biologists, tend to study land plants and well, vertebrates. I mean, there's quite a bit of invertebrate biology as well, but we're really studying only a very small portion of the entire tree of biological diversity. Most of biological research is based on a very small fraction of the tree of life.
Hmm. And so because it's so hard to cultivate and to just find life forms that are out there, there's, probably a whole bunch of stuff we haven't discovered. Do we have any sense for how much of the tree of life we've actually discovered and how much is out there to be discovered?
Yeah, it's a real good question. Um, I mean, this whole idea of us being derived archaea has only come about in the last 10 years. And that was a consequence of sampling some, deep sea samples, just doing what's called metagenomic sequencing. So just taking a lot of DNA out of a bottle of mud and then, sequencing to a high enough depth that you can actually start to construct the organisms that are down there, even the absence of any visual data on them. But, that's just one, you know, semi unusual environment. There's almost certainly other, uh, maybe very substantial discoveries to be made. We still have uncertainty about what particular branch in the archaea eukaryotes came out of for this very reason. And we may eventually find particular lineages that are, um, intermediate states between current archaea and eukaryotes. So this is the problem we have right now is most eukaryotic lineage that we've looked at, like everything was there. So mitosis was there and internal organelles and so on. And so there's this big morphological gap between us and archaea, but it's not to say there aren't some deep archaeal lineages with intermediate phenotypes that might then help us understand the order of events in the emergence of eukaryotes.
Hmm. Going back even deeper, when we think about, so when we look at the diversity of life that we have uncovered, and we think about the origin of life, do we know pretty definitively there was one origin of life on Earth, or what do we actually know about the origin of life? Is that something we can figure out? Is it something that we know happened once, or at least once? Is it possible that multiple origins of life happened, and then there was some kind of mixing of them? What do we know about that root of the tree of life?
Well, there's a lot of things to unpack there. Um, we think that all current lineages that we're aware of go back to a single origin. That's based, though, on a very small number of genes shared, less than 100 genes that are shared among all organisms. We need those genes they provide the information on developing genealogies of shared and unique differences. So there's not a lot to go on there. There are a number of other issues here. One is that the way we replicate DNA, archaea and us derived eukaryotes replicate DNA is using machinery quite different than bacterial lineages use. So that's led Eugene Koonin and colleagues to suggest maybe replication mechanisms in these two lineages today evolved independently. There very well could have been many other independent lineages of life that were going on at the time. There's this thing that we call LUCA, the last universal common ancestor. That's the ancestor to all of us living organisms on Earth. Could very well have been other lineages that were around at the time that went extinct. It's unlikely we're ever going to be able to resolve that because DNA, the remnants, the molecular fossils we need are just not going to be there. We can't even formally rule out the possibility that life originated on another planet and was transmitted here by a comet or so. So we know that based on phylogenetic information using molecular fossils and gene genealogies, that life seems to have started very soon after the Earth originated. Getting very, very close, back to 4 billion years. So things happened pretty quickly. And of course, at that time, the earliest forms of life would have been pre-cellular. We're going to have no information on that. There's this whole hypothesis about an RNA world. Carl Woese had this idea that the earliest form of life was sort of almost a kumbaya sort of community in which you had all kinds of, became very unclear what an organism even was under his view and what a population was and so on. But then eventually cellularity took hold. And now we've got independent lineages where individual genotypes are linked inside internal cell membranes. So there's a direct connection now between genotype and phenotype and inheritance from parent to offspring cell.
Mm-hmm. What's the best estimate for when the first cell evolved?
Well, the latest estimates I was alluding to, again, this is based on shared inheritance of this small number of genes that we all have, and making assumptions about what a thing we call the molecular clock.
Mm-hmm.
So we can measure divergence among phylogenetic lineages. We can do that. There's X% divergence, but we need to know how to translate that into time. Mm-hmm. How many changes occur per nucleotide a site in a genome per unit time. So we have to use these calibrated molecular clocks. The latest estimates are pushing the root of the tree of life back to about 4.1 billion years. So this is getting very tight because we think the Earth originated about 4.4 billion years.
So is it fair to say that over time, our understanding of our estimates of the origins of life have gotten closer and closer to the origins of the Earth? In other words, that life emerged more quickly than we used to think.
Yeah, these molecular estimates are pushing things back farther. There are quite convincing fossils. We don't know what they are, but we're quite convinced they're biological. Certainly go back to 3.8 billion. So that much we're sure of. But now the molecular data are pushing things back even further.
And, going back, so you said there's these about 100 genes that are sort of common to all life forms. What are those genes encoding? That's related to the question of what are sort of the universal features that all forms of cellular life share? What are the structures and the cellular functions that they're all doing that are that core set of things?
Well, the core set of biology, what is biology made of? What's the basic biology all organisms have is pretty broad. But over time, there have been what we call non-orthologous gene replacements and so on. So the same function that's in a certain lineage won't necessarily be carried out by the same gene.
Mm.
And there have been probably de novo origins of different genes and so on. So there are a lot of functions that we share, things like, the Krebs cycle. We all make lipids to produce membranes. Glycolysis, so things that go back very deep. All the translation machinery, as I just pointed out, the replication machinery is quite different. The translation machinery, the ribosome, that goes back all the way. That's why Carl was using ribosomal RNA to really look at the entire tree of life. The ribosome itself is a bizarrely complex organelle. There depends on what lineage, but there's on the order of 100 proteins wrapped around the ribosomal RNA. Some of those proteins seem to be shared by everybody.
So this is the protein structure in cells that takes genetic material and transforms it into proteins. And
correct.
Parts of this go back all the way as far as we can tell. That's what you're saying.
Yeah. Yeah. So we really have, there's some bizarre features of biology. We have genomes made of DNA. That's where the messages are. That's where the vocabulary is to produce proteins. But the DNA has to be transcribed into these things we call messenger RNAs. That's RNA. The RNA then has to be read by the ribosomes that translates codons, which are each 3 nucleotides long. Each one stands for a particular amino acid. And then there's a stop codon. 3 of them are stop codons. 3 of the triplets are stops. The ribosome works together with things called transfer RNAs that bring in an amino acid that then links to it has an anti-codon that then reads off the codon. So all biology has tRNAs and ribosomes. So those are an example of things that go all the way back.
And I suppose that's why people have ideas about this RNA world, that RNA and proteins were there before DNA came into the mix.
Well, that's a good question. Did proteins come before DNA or vice versa? There's a strong feeling that RNAs had to have been around early on. The fact that transfer RNAs are made out of RNA and ribosomes are made out of RNA suggests that there must have been some code early on. Whether that code itself, the genome itself that was being read off was also RNA, and then there was later DNA takeover is a bit unclear. But given there was a code, then seems likely there were amino acids doing something, right? Because the code is for the readout of amino acids. Whether they were being read out to make the kinds of complex proteins that we have today is another question as well.